In this lecture, we're going to talk about genetics, and we're going to start from a historical perspective and look at the Gregor Mendel, who is considered the father of genetics, and some of his contributions to our current day knowledge of heredity. So in this picture, we see Gregor Mendel, who was a monk. Here he is, holding a fuchsia sprig. He was a monk in a monastery in current-day Czech Republic, and he was really a true scientist in that he believed in evidence-based science. He actually performed experiments that led to what are called the first and second laws of genetics, and he did this by working with pea plants. And I might add that this is back in the mid 1800s, and at that time it was known that there are certain traits that are passed on from parents to offspring, through the generations in plants and animals, but the current hypothesis was something they called the blending hypothesis, where the hereditary information that you received was sort of mixed, a mixture of your parents' hereditary units, and sort of like yellow and blue paint mixing to form green paint. So they call it the blending hypothesis. Now, there were a couple problems with this hypothesis. It didn't explain why, as the generations progressed, the individuals of that species didn't become more and more alike as their genetic material was mixing. It also didn't really explain how genetic traits could disappear for generations and then reappear. So Mendel's hypothesis was quite different than this blending hypothesis. He proposed that these genetic hereditary units remain discrete and were passed through the generations, sort of like shuffling cards in a deck. So let's look at some of his early famous experiments. So he worked with the pea plants, which were easy to work with, and they had some very distinguishable hereditary traits, like the color of their flowers. That was easy to see the difference. So he took what are called true breeding parent plants. True breeding means that the purple, this purple flowered plant right here, all of its offspring will be purple. It's a true breeder. It always produces purple offspring. And the same with the white flowers. They were also true breeders. All their offspring had white flowers. So he took those and he cross-pollinated them, and the offspring, what we call the F1 generation, the first filial generation, are what are called hybrids, meaning one parent, their two parents were different. One had purple flowers, one had white. And notice that all the offspring were purple. Okay, but are they just like the parent plant, genetically speaking? Well, to help answer that, he crossed two of these F1 plants together and found that in the F2 generation, if both the parents were hybrids instead of true breeders, then look at what happens here. Most of them are still purple, but a few, or a smaller percentage, of the offspring are white. So this is what I meant by, Mendel meant by discrete inheritable units of heredity, that this white flower hereditary unit is not lost in the F1 generation, but it is masked, and then it can reappear in later generations. Okay, this also led to another concept that some hereditary units can mask others. So if you're talking about flower color, the purple hereditary unit will mask the white hereditary unit. And we know that from Mendel's experiments, seeing that all the F1 are purple. And yet they have a purple hereditary unit and a white hereditary unit, one from each parent. So we say that the purple is dominant to the white hereditary unit. It is dominant. And usually the dominant hereditary unit is symbolized with a letter and a capital letter. So for example, we would usually use a capital P for purple, for the purple hereditary unit, and for the white we're going to use the same letter but lowercase. So like lowercase P would stand for the white flowers. It helps to use the same letters here, big P and little p, because we are talking about the same hereditary unit, flower color. So some vocabulary. There's a lot of vocabulary in genetics. I highly encourage you to become comfortable with these terms. So these basic units of heredity that Mendel was talking about, we now call genes. Okay, he didn't use that term, but that's what we call them now, these hereditary units. The alternate forms of a given gene are called alleles. So alleles are alternate forms of a gene. So for example, the purple and the white hereditary units, those are alleles, and therefore the gene for flower color. So alleles are alternate forms of a gene, like purple and white. Okay, now jumping ahead into the, oh, well, not yet. This slide shows some of the other traits that Mendel looked at. So he was lucky that he chose the pea plant because it has a lot of easy to follow inheritable traits that follow a dominant pattern where one allele is dominant over the other. The allele that's not dominant or is dominated by the other one is called the recessive allele. So again, using purple and white, purple's the dominant allele, white is the recessive allele. And that's why the F1s were all purple, because even though they had one of each, purple was dominant. And there were many other traits you can see here that he looked at, like seed color, yellow or green. Is the seed round or wrinkled? He looked at pod shape and color, stem length, and so on. So it wasn't just flower color that he was following. Now, to jump ahead for a moment and going back to what we learned about meiosis, remember that because, well, to keep this in perspective, Mendel did not know about meiosis. Meiosis was first described in the 1880s, so I guess during his later lifetime, but meiosis was not a well-known concept, nor were chromosomes. It wasn't until the 1900s that they came up with something called the chromosomal theory that we'll talk about in our next lecture, which just states that our hereditary material is located on chromosomes. I mean, this is something we know now, it's common knowledge, but I just wanted to make sure you understand that in Mendel's time, they didn't even know about chromosomes, and they definitely didn't know that on the chromosomes it was the DNA that was actually the structure of these hereditary units. I mean, that wasn't until the 1950s. So more on that later. But since you have learned about meiosis, let's just look at where the alleles are located. Remember, we learned that homologous chromosomes contain genetic information for similar traits. So now we can use the vocabulary that we just learned, alleles, and we can say that homologous chromosomes contain alleles for the same genes. And not only that, but notice they're in the same location or locus on the chromosome. So that's really what it means to be a homologous pair of chromosomes, for the most part, unless you're talking about the X and the Y chromosome. But because in general, they contain alleles for the same genes in the same locations. Okay, some more vocabulary. If the organism, in this case pea plant, has if both of its alleles for some trait like flower color are the same, like if it has two purple alleles, then we say that that organism is homozygous for that trait. Homozygous. The two alleles are the same, two purple alleles. If they're different from each other, like if the plant has a purple and a white, remember the lowercase p is the white allele. If it has one of each, it's called heterozygous. Okay, heterozygous. And then down here, if we look at white, this is homozygous. Homozygous because both alleles are the same. And to more fully describe these, we could say this plant right here is homozygous recessive. It has two recessive alleles for flower color. The one up here at the top, this is homozygous dominant. It has two dominant alleles. Okay, and then also some more vocabulary. Look up here at the top. You see this term genotype? Just break down the word. Type of genes, genotype. It really means specifying the two alleles for that particular gene, like flower color. Remember, that's the gene we're talking about right here, flower color. So the genotype is literally, it's describing what are the two alleles. So the genotype for this flower, or this flower here in the middle, is big P, little p. Now phenotype has a different meaning. Phenotype, it means the physical manifestation of that genotype. So like, what is the physical effect of that genotype on the plant? So for flower color, you're going to give the color of the flower. So the big P genotype has a phenotype of purple flowers. But so does the heterozygous plant. It also has a purple phenotype. Whereas to be white, to have a white phenotype, you have to have this genotype, little p. Because even if you have one purple allele, it'll dominate and the flowers will be purple. So notice that two different plants can have the same phenotype, purple, but different genotypes. They can be homozygous or heterozygous. So there's some more vocabulary for you. Definitely want to get this down so that you don't have to even think about when you read these terms, because they'll come up over and over again. Okay, so just to look again at this experiment. So he's taking, so now we've got the genotypes in there. So when he crossed the true breeding purple and the true breeding white flowers, let's try to think about what's going on during the meiosis and reproduction here. Remember, in meiosis, the daughter cells, which we call gametes, are haploid. They have only one of each homologous chromosome, and that means that they only get one of the two alleles for a given gene. So if you need to review meiosis, please do so. It's going to help you a lot with genetics. So the gametes, these would be the daughter cells from meiosis. The gametes of the true breeding purple flower are all going to be the same. They're all going to carry the purple allele, and the same with the white flower. All of their gametes are going to have one white allele inside the gamete. Okay, so when those gametes fuse during fertilization to restore the diploid number, the full chromosome number, to restore the homologous pairs, all the F1 got a purple allele from the purple flower parent and a white allele from the white flower parent, and so their genotype is big P, little p, heterozygous. That's their genotype. What is their phenotype? Purple. They're all purple. Now let's look at the kinds of gametes this plant can make. They can actually make two different kinds of gametes. Half of them will contain the purple allele and half the white allele. So look carefully at what's going to happen when these F1 plants, when you cross two F1s. In other words, where one parent is big P, little p, and then you're going to cross it with or mate it with, cross-pollinate it with another plant that has that's heterozygous for flower color. So here's how you set up this kind of problem, and you're going to be using this a lot in doing genetics problems. So let's take a minute here and look at how you figure out not only what kind of F2 plants genotypically and phenotypically will be formed by crossing two F1s, you know, big P and a little p with another big P, little p. You set up what's called a Punnett square. So this is called a Punnett square. Very helpful in genetics. Very powerful tool to use. And pretty simple once you learn how to set it up. So for a Punnett square, what you want to do is you're going to draw a box, and you're going to put across the top, you're going to put all the different kinds of gametes that the male plant can make, and then down the side the same with the eggs, or it can be switched. It actually doesn't matter. You can have the eggs across the top, sperm down the side, just so you have one parent across the top, the other parent down the side. And then you need to for each, you need to make enough boxes so that you have all the different possible alleles for that parent written in your Punnett square. So notice we had to have two here, because there's the capital, because the F1s have two kinds of alleles, purple and white. They carry both. So half the sperm are going to be purple, well, they're going to carry the purple allele, and half white. And then for the female, same thing, because it's heterozygous, half purple, half white. So you write those different alleles down the side. Okay, now this is usually what students have the most trouble with setting up their Punnett square, is making sure they get all the different possible gametes. Once you get that, it's pretty easy to then fill in your Punnett square. But before we get to that, let me just back up for a moment and let's look at the original parents up here. If you were doing a Punnett square for this cross up here, for the purple and the white, it's actually, very simple Punnett square, in that the purple flowers can only make gametes with the purple allele, and the white flowers can only make gametes with the white allele. That's it. Those are the only possibilities. I mean, you can list it multiple times and make a bigger Punnett square, but you really only need the different possible gametes across the top and down the side for the two sexes. That's really all you need. So this is the minimal way to do it. And then you say to yourself, well, what's going to happen if the purple allele and the white allele if these two gametes are fertilized and form a new offspring, then what's its genotype going to be? It's going to be big P, little p. Right? It's going to get one allele from one parent, one from the other. I mean, this is what meiosis and fertilization is all about, sexual reproduction. So notice that all the offspring, all the F1 are going to be big P, little p. This is their offspring. So again, these are the gametes from the original parents, and then inside the box is the genotype of the offspring, or what they call the F1. What is their phenotype? Purple. They're going to have purple flowers because purple is dominant to white. Okay, so back down here to the F2. So this one's a little more complicated. You need a bigger Punnett square because, again, the parents are heterozygous, not homozygous, so they can make more than one kind of gamete. So again, purple and white in both sperm and eggs. And then you just fill in. So once you set up the Punnett square, you just fill in the boxes. The boxes represent the possible offspring that they can produce. So if this purple sperm and that purple egg are fertilized, then the new offspring has a genotype big P, and it'll have purple flowers. In this box here, you consider what would happen if this purple egg and this white sperm fertilized. Then you get big P, little p. It's still going to be purple though. And then again here, big P, little p. Little p, little p. So that's how you fill in the Punnett square. And what it's telling you is what are the possible offspring that these two parents can have. If both parents are heterozygous and purple is dominant to white, what are their possible offspring? It also tells you something else. It also gives you some ability to predict the chance of various genotypes or phenotypes. So if you look at inside the boxes, you notice that 3 out of 4 are purple, right? There's 4 boxes in here. 3 out of 4 are purple. So there's a 3 out of 4 of the offspring there's a chance of their probability that those will be purple, or you could often they use a percent. So you could say there's a 75% chance that these two plants, heterozygous plants, will produce purple offspring. And then what about the white? Well, notice it's 1 out of 4 are white, so that's 25% chance that you'll get white offspring. Now that doesn't mean that if you cross these plants and you get 4 offspring, 3 will definitely be purple and 1 will be white. This is a probability we're talking about here. Just like flipping a penny, heads or tails. It's a 50-50 chance, but if you do it 100 times, you're probably not going to get exactly 50 heads and 50 tails. You know, this is a probability. Still, it's informative and helpful to look at. So that's what Mendel did in his early pea experiments. Okay, and just to practice some more with Punnett squares, let's look at a case where you have some unknown, you have a purple pea plant. Let's say Mendel found one of these and he didn't know, oh, is this heterozygous or homozygous? So it's not like you can look in there and see. So one thing you can do is what they call a test cross. So he could cross this purple flower with its unknown genotype, because it could be big P or big P, little p. They're both purple. So if you don't know, what you can do is cross it, mate it with a white flower, because for white, you know the genotype. There is only one genotype for white flowers, since it's a recessive trait, little p, little p. So you set up your Punnett square. You've got your sperm across the top, little p, little p. Notice they did it twice. You could just do it once. I mean, really, you just have to list the different possible ones. But this is fine here. It'll work out either way. So the gametes are all going to have the white allele in it. And then he set up we could set up 2 different Punnett squares. We can say, well, what if the purple flower is big P? Then all the eggs are going to be purple, have the purple allele. And then if you fill in the Punnett square, big P, little p, big P, little p, big P, little p, big P, little p, they're all the same, right? So you would get all the offspring are expected to be purple, no whites. On the other hand, what if the purple plant was actually heterozygous. Then you would get half purple allele-bearing eggs and half white. The sperm is going to be the same across the top. Fill in your Punnett square: big P, little p, little p, little p, big P, little p, little p, little p. And notice that in this case, the expected offspring is 2 out of 4, or 1/2, or 50% white, and 2 out of 4, 1/2, or 50% purple. So half and half. So then you do the cross, and then you look at the offspring. Are they all purple, or are there some about half white? And that's how you could tell what the genotype of this original purple flower is. So Mendel's work looking at 2 alleles for a heritable trait allowed him to come up with what is now called Mendel's law of segregation. So this is a basic principle of genetics that every biology student learns. And it just states that the 2 alleles for a given gene or heritable trait, like flower color, are going to separate during gamete formation and end up in separate gametes. In other words, each gamete just has one of the 2 alleles that were in the parent, the parent that, or the plant that's producing the gametes. Their gametes are going to have just one of the 2 alleles for a given gene or trait. Okay, now this should make a lot of sense if you look at meiosis, because that's what's happening. So just looking at this once again, notice, these could be, here's the purple flower allele, and here's the white flower allele. They're on homologous chromosomes, and remember, that's the whole purpose of meiosis, especially stage 1 of meiosis. That's when the homologous chromosomes synapse. There might be some crossing over, but then they separate during anaphase 1 of meiosis, and in the end they end up in separate gametes. So that's Mendel's law of segregation. Okay, and here again I just want to remind you about homologous chromosomes. So these are homologous pairs here. So again, they contain alleles for the same genetic traits: eye color, blood type, whether or not you have the enzyme to digest lactose, things like that. Anything that's a genetic trait, the genes for those are going to be carried on these homologous chromosomes. Okay, now there's another law of called Mendel's second law, the law of independent assortment, and this is also more easily understood if you know your meiosis. Okay, and in the law of independent assortment, Mendel had to look at what would happen if he looked at 2 different genes, 2 different hereditary units with 2 different pairs of alleles. And he found that if you're that each pair of allele segregates independently of the others during gamete formation. So again, this goes back to meiosis. So what we're talking about is 2 different pairs of alleles. So let's say flower color and seed shape. Like whether a plant has purple colors and round seeds, or white colors and flowers and wrinkled seeds. Those are 2 different genetic traits. They each have a pair of alleles. And so what Mendel is saying is that those 2 different pairs assort independently of each other. Like the purple and the round seeds may go into the same gamete, or they, if the plant also has a wrinkled allele, it might go into the same gamete as the purple. It's a matter of probability, and this all goes back to something we talked about in the meiosis lecture. How during meiosis 1, the metaphase 1 of meiosis, the non-homologous chromosomes. Those are depicted here as the big and little. The non-homologous chromosomes, the way they line up on the metaphase plate is random. So like on one side you see the blues are on one side and the reds on the other. On possibility 2 you see there's a mix. Okay, that's random. But if you think about what's on those chromosomes, maybe the big ones carry the flower color and the small chromosomes might have the allele for the round or wrinkled seeds. So depending on how these chromosomes line up on the metaphase plate, they may or may not go into the same gamete. So Mendel picked 2 of these traits, 2 different ones, and followed them both during in some of his experiments. So here's his basic. This is called a dihybrid cross. Okay, and you're going to be doing some of these too yourself, where you look at 2 traits instead of just 1. Okay, and you can also look at 3 traits or more. We don't in Biology 150, but if you take genetics, you probably will. So in this example, we're looking at the parent generation. We're looking at some true breeders, some homozygous plants. So in this one, this pea plant, their seeds are yellow. So capital Y is for yellow. And they use the capital Y because it's dominant. It's the dominant trait. So little y is for green. And again, don't use the little g. You want to use, these are alleles for the same trait, seed color. Therefore, use the same letter if it's a dominant recessive situation. And for the dominant, you always use the capital letter, lowercase letter for the recessive allele. And I highly recommend when you're doing genetics experiments that you write out your key over here so you don't get mixed up. Okay, and then notice they're using capital R for round because that's dominant, and then lowercase r for wrinkled. So that's the parent generation. Okay, and then if you do your Punnett square, it's going to be a simple one, but you have, well, look at the gametes though. The gametes are going to have 2 alleles in them, one for color and one for shape. So they're going to have the yellow, so like this plant, the yellow round seeded parents, there's only one kind of gamete they can make. They'll all have the yellow color and the round allele. And then the same for the green wrinkled true breeding plant. All their gametes are going to have the green wrinkled alleles in them. I mean, that's all they have. So if these undergo fertilization, then all the F1 are going to be heterozygous for both traits. Big Y, little y is for color, and they're going to be yellow, as you see here. Big R, little r, they're going to be round, again you see in the picture, because yellow and round are dominant to green and wrinkled. So there's all the F1. Okay, they look like, the F1 look like the yellow round parent, but notice they're not genotypically the same. They are phenotypically the same, but not, they have a different genotype. So Mendel, here was Mendel's question. When he crossed the 2 F1s, so one of these fertilizing another with the same genotype, are the alleles from the original parents going to stay together? In other words, are the yellow and the round alleles going to go together into the offspring and during fertilization? Will they be assorted together? And the same with the green wrinkled? Or can they assort independently? So in other words, can you have, yes, you'll have some yellow and round alleles in the same gamete, but can you also have some yellow wrinkled or some green round and green wrinkled? So that was the question he asked. Will the alleles stay together or assort independently if they came from the same parent? So he just set up the Punnett squares and filled it in and looked at the possible outcomes. So if the parental alleles stay together, these are the different sperm and egg possibilities that you see here. And again, it's just like the previous one we did, which is called a monohybrid cross. We're just following one trait. This is a dihybrid cross. It's the same principle. So you've got these two gametes. If they fertilize, then you restore the diploid number. You get big Y, big Y, big R, big R. Or down here, you would get little y, little r, and another little y, little r, and here's the, oops, sorry, it's messy, genotype. Okay, I highly recommend you keep your alleles together. In other words, don't write YR, YR. Keep the Y's and the R's together. It'll help you analyze the results more easily. Okay, so if the alleles segregate together, then notice that 3 out of 4, or 3/4, are going to be round and yellow, and only 1/4 green and wrinkled. But over here on the right, you see that if the alleles can segregate independently, and you can get like a color from one parent with the shape from the other, so that you could have conceivably green wrinkled or yellow, green yellow, green round offspring. Then it's a little more complicated, because across the top, notice there's 4 different kinds of gametes: big Y, big R, big Y, little r, etc. And the same with the eggs, because they're heterozygous for both traits. So make sure you have all the different possible gametes across the top and down the side. Okay, and so it usually helps to write out what your cross is. Like you have big Y, little y, oops, and then big R, little r. If you want to figure out all the different possible gametes from this, they usually use the FOIL. You can use the FOIL method, where you take the first of each. So you have like big Y, big R, and then you take the outer ones, big Y, little r, and then you take the inner, little y, big R, and then the last of each, little y, little r, to get all 4 types. Okay, you don't have to use the FOIL method, but you use whatever method works for you, but just make sure you get all the different possible gametes down the across the top and down the side. You need them all, because that's going to show you all the different possible offspring. And then you just start filling in your Punnett square. So this is a big 16-box Punnett square. For our class, this is the biggest you'll be doing, because we don't go beyond dihybrid crosses. So you just fill it in, looking at if this egg and that sperm fertilize, these are the different possible offspring. So you just fill them all in, then you have to go through and analyze them. How many of these are going to be yellow and round? So you got to look at their genotypes. And you see that 9 out of 16 are yellow and round. And then you count up, how many are green and round? There's 3 out of 16. How many are yellow and wrinkled? 3 out of 16. And then only 1 out of 16 is, I think I might just said the wrong thing, is green and wrinkled. Okay, 3 out of 16 yellow and wrinkled, 1 out of 16 green and wrinkled. So those are the phenotypes. So it's a 9 to 3 to 3 to 1 ratio, or 9 out of 16, 3 out of 16, and so on. Or you could do percents if you prefer. So this is what Mendel was thinking, that these would be the different expected phenotypic ratios if the alleles did not, or if they did segregate independently. So he thought about that, then he actually did the experiment, and these are the results he got. His phenotypic ratio, so these were the actual numbers from his pea plant crossings, and you see that it approximated, it's never going to be exact, but it approximated 9 to 3 to 3 to 1 ratio, and so that supported the this idea of independent assortment of allele pairs of different for different genes. Okay, so once again, it has to do with meiosis 1, or metaphase 1 during meiosis, when all the homologous chromosomes line up on the equator. Now, in our later lecture, in our next lecture, we will talk about how if two different genes are very close together on the same chromosome, are they going to end up in separate gametes? We call those genes that are very close together on the same chromosome linked genes. And we will talk about this more later, but they will be more likely to go together into the same gamete. But if you recall, we did talk about something called crossing over in meiosis 1, prophase 1 of meiosis. And so, because of crossing over where the homologous chromosomes can swap some genetic information, they could still end up in separate gametes. So it's not impossible, just not as likely. Okay, now not all alleles are simple, complete, dominant, or recessive alleles. There's also something called incomplete dominance, where the phenotype of the heterozygous F1s is somewhere in between the parents. So we'll look at that in a moment. And then we'll also look at something called codominance, where both alleles are equally expressed in the phenotype. So here's an example of incomplete dominance. So notice the parents are red. This one parent is red, one is white. And now they wrote their genotypes a little differently. They're using a big C for color, and then a little r for red, and a little w for white. Okay, so there's different ways to symbolize the alleles. Anyway, both are homozygous, right? 2 red alleles for the red, 2 white alleles for the white plant, and they can each only make one kind of gamete, and all the offspring are going to be heterozygous. They're going to have a red and a white allele, but notice that this is not a case of dominant and recessive. If it was and red was dominant, then these would be red, just like with the purple pea plants. But instead, notice their phenotype is in between. It's pink. So the heterozygote that has a red and a white allele has a phenotype that's somewhere in between the two parents. Okay, now this kind of seems to support the blending hypothesis, but it actually doesn't. Because if you take 2 of these pink alleles, 2 of these pink snapdragons, these are snapdragons, and cross them, in other words, 2 pinks. So notice, setting up your Punnett square, if they're heterozygotes, they're going to make 2 different kinds of gametes. Half will have the red, half will have the white allele. Same with the other plant, because we're crossing 2 pinks. And then you fill in your Punnett square, and then go through and analyze, and notice that there's so the kinds of questions I might ask you in a genetics problems are, if you cross 2 pink snapdragons that display incomplete dominance, what's the chance they'll have red, they'll produce red flowered offspring? So you'd say, well, it's 1 out of 4, or there's a 25% chance they'll have red flowers. What about pink flowers? You say, well, out of 4 boxes, there's 2 are pink. So 2 out of 4, or 1/2, or 50% will be pink, most probably. And then what about white? 1/4, like the red, 1/4 or 25%. And by the way, your percents all have to add up to 100. So we're saying there's a 25% chance red, 50% chance pink, 25% chance white. That adds up to 100%, all the offspring. So notice this does not support the blending hypothesis, because notice the white and the red alleles, both of which were not evident in the F1 generation, have reappeared. Okay, so that's incomplete dominance. Now what about codominance? Codominance is when both the alleles are equally expressed, both. And we see this in blood type. So just quickly to give you a quick overview of blood type. On our red blood cells, we have markers that help our immune system know whether they belong to our body or not. And there's a bunch of different kinds, but the ones that we're going to talk about here are called the A and the B markers. The A and the B markers. Okay, and they're important because they're well, we won't go into it too much, but we have antibodies against the A or B marker if we don't ourselves have them. So it matters what your blood type is. If you're going to get like a blood transfusion, for example, you have to have compatible blood. Anyway, there's two kinds of markers then, the A and the B markers. These are determined, but you have alleles for them in your on your pair, one of your homologous chromosome pairs. So you have one allele, from your mom and one from your dad. So let's say you inherit an A allele from your mother and a B allele from your father. And with blood type, this is the common way they write it, with a big I and then a superscript, A or B. So if you get an A allele from one parent and a B allele from another, they are equally expressed. In other words, you are going to make both A and B markers on your red blood cells. And so we say that neither one is dominant or recessive to the other. We say that they are co-dominant. They are both expressed equally. And we would say that your blood type is AB, type AB. Now, if you only have the A marker, not the B, then your blood type is A. If you only have the B marker, not the A, then your blood type is B. And there is a third one, because blood type also points out another feature about hereditary units, and that is that there's not necessarily just two different forms of an allele. There can be multiple alleles for a specific gene. Now, I mean, even though you only have two alleles for that gene, one from your mom, one from your dad, out there in the population, there can be 3 or 5 or more different variations, 2 of which you will get, depending on your parents. So for blood type, there is a third allele. It's designated by a lowercase I. They use the lowercase because it is recessive to the A and the B, and this is the little I is for O, stands for the O, type O blood. Type O blood actually means you don't have the A or the B. It's not like there's a third marker called the O marker. It's that you don't have the A or the B, you're type O. So they use a lowercase because it's recessive to the other two. So let me just review this again. So the A and the B alleles are co-dominant. They are equally expressed if you get both. And then the O allele, designated by lowercase I, is recessive to the A and the B alleles. So if your genotype, if you got a B allele, say from your mom, and an I allele from your dad, your phenotype is going to be type B blood, because B is dominant to the O allele. The only way to have type O blood is to have two O alleles, 2 of the little I's. So type O blood is a homozygous genotype. So blood type is very popular to use in genetics problems. I'm sure you'll have one coming up soon. Okay, and then here's another term that you need to at least know the definition: pleiotropy. Pleiotropy means that sometimes a gene can have multiple phenotypic effects. So we've been sticking to simple things like blood type and flower color and seed shape. But there's also gene alleles that have multiple effects on the phenotype. So for example, some genetic diseases, which we're going to talk about in a minute, they could be due to just a single pair of alleles, as they are in sickle cell anemia, but they can have multiple effects on the individual that has them. They can affect many different things. Or another example would be cystic fibrosis. It affects not just your respiratory system, but also the digestive system. So that's what pleiotrophy means. One gene having multiple effects. Okay, here's a slide that reviews the blood types. So kind of backing up here. So these are the 4 ABO blood group types. So you're one of those 4. You're type A, you're type B, AB, or O. And this shows the possible genotypes for those. So notice there's only one possible genotype for type AB. I mean, if you know your type AB blood, you know your genotype. There's only one possible genotype. You had to get an A from one parent and a B from the other. Same with O. If you're type O, you know your genotype. But what if you're type A or type B? Then, you can't be sure whether you're homozygous or heterozygous, because O is recessive to A or B. Okay, and then here's a definition of pleiotrophy. So we already talked about that. Let's skip through that. Here's another, yet more vocabulary. Polygenic inheritance. What does that mean? Well, just look at the term. Poly means many genes, inheritance. So what it basically means is that there are some traits that are determined, that have a strong genetic component to them, like how tall you are. I mean, your environment also matters. We'll talk about that in a minute. But, there's a strong genetic component to your height. But it's not like there's just one pair of alleles that determine that, like a height gene. It's instead due to many alleles collectively. So that's what polygenic means. Traits like height that are a result of many alleles. All right, so here is another concept that you need to know in general what it means, although you won't be actually working a genetic problem with epistasis, but you might be asked what it means. So epistasis is when a gene at one location on a chromosome affects the expression of a gene, a separate gene at a different location. So here's a good example with mice coat color. Okay, so for coat color we're going to use B, the B as the symbol. So big B, and remember, I highly recommend you do this for every genetics problem you do, so you don't get mixed up. So big B is for black. Okay, now how would you know that? You would have to be given some information in the problem, like they'd say black coat color is dominant to brown. So we're going to use little b for brown. Or they might not tell you that directly. They might say heterozygous mice have a black phenotype or something. That's how you figure out. You have to be given information in these genetics problems. You're not expected to know which genes are dominant or codominant or incompletely dominant and so on. So you're going to be given information to figure it out. So anyway, in this problem, black is dominant to brown coat color. But then look at these crosses of mice where they're crossing they're actually crossing two black heterozygous mice. And so you would expect that in that case you would get some black and some brown mice, not white. We'll get to that in a minute. But so you would expect they would either be black or brown. And if you look down here at the genotypes, you see that some of them conform. Like here's a little b genotype in this mouse, and it is brown. But But then look over here. This mouse also, oh, well, that's brown. Whoops. Okay, look down here at this mouse, down here at the bottom. It also has 2 brown alleles, but why is it white? Because in this example, there is another set of, another gene, we're going to call it C, and there's big C is, let's say it's for a functional, an enzyme or that's needed to deposit. This will add the color to the coat. So it's like an enzyme that the mouse needs to actually produce the pigment that it has the alleles for. And then lowercase c means it does not, that allele does not add the pigment to the coat color of the mouse. So the whole point of this is to point out that it's not just whether you have the alleles for a certain trait like coat color. Sometimes it also matters if you have another pair of alleles that's needed for the expression of that first pair. So if the mouse doesn't have the, let's say it's an enzyme, okay? If it doesn't have the enzyme, like let's look down here at this white mouse. Notice it has 2 lowercase c's. That means it lacks the enzyme. So even though it has the brown allele genetically in its genotype, it cannot properly insert that pigment into the coat color. So the mouse looks white. It's an albino mouse, or white, it lacks coat color. Okay, now if we look over up here at this mouse, it also has the brown alleles, but notice even though it also has a defective allele, we could call it, it does have one capital C, it does have at least one allele that would allow it to put the pigment into the coat. So that's enough. So it does express that coat color. So that's what epistasis means. It means a second pair of alleles affects the phenotypic expression of another pair of alleles. So it's not enough just to have, you know, the black allele to have a black coat. You also have to have the, like look at this mouse right here that I'm circling. It's homozygous for black coat color, but it's white because it doesn't have that functional enzyme. Now, I don't want to underplay this, even though I'm not going to go into it too much, but you know, the genes that you inherit from your parents do not determine solely your phenotype. There is also the effect of the environment, and it's a big effect. So this is the old nature versus nurture. You know, are you a product of your genetic makeup or of the environment that you grow up in and live in? And the answer is both. It's both. So here's a great example, kind of simple example of how the environment can affect the phenotype of, in this case, hydrangea flowers. So these two plants, or actually let's say it's the same plant, right? It's the same plant. So it's got the same genes, but in one picture the flowers are pink and the other they're blue. So what's going on here? Because it's not a genetic difference. Same gene, same alleles. What's different is the environment. For the pink flowers are in a different environment. They like a basic pH. Whereas the blue, if the pH is more acid and you add some aluminum to the soil, then the flowers will tend to be blue instead of pink. So that's just showing the effect of the environment on the phenotype. It's not just the alleles. Or here's another good example, how temperature can affect the coat color in certain animals. Like in the Siamese cat, why is the tail and the tips of their feet or the tips of the ears black, and whereas the main body is white? It's not that there's different coat color alleles there. It's a temperature thing. So the temperature is cooler in the tail and the extremities than in the main body, and that affects the, in this case, expression of coat color. So the environment plays a big role. Okay, now pedigrees are something that we use with humans a lot, because you can't, like pea plants, be mating humans and doing experiments with them. So this is not a formal part of our course. I'm not going to spend much time on it, but I am going to tell you that there might be, hint hint, a bonus question on your next test using pedigree analysis, and it's definitely something you'll cover in genetics. So this is going to be a quick example, and you can read about it in your book. With pedigrees, you look at generations. So let's just look at one. So we're looking at sisters, and then here's their parents and their parents' siblings and their grandparents and so on. And in general, squares are males, circles are females, and if they're colored in, like in this case in purple, it means they have that trait, or sometimes it's a disease you're following. So in this case, it's not really that exciting of a trait. It's this widow's peak. If your hairline forms a V versus not, they call that a widow's peak or non-widow's peak. And it's a genetic, it's a dominant trait. So capital W is for widow's peak. To have the non-widow's peak, you have to be recessive, little w. So this just allows you to kind of follow through the generations different traits. So you see, if these two sisters, if you know that widow's peak is a dominant trait, then you know her genotype, the one without it, because it's homozygous recessive. But for the other sister, you're not sure whether she's homozygous or heterozygous. You can't just tell from that. Then you could try to figure it out by looking at her parents and their parents and so on. So, that's the basis for pedigree analysis, is that you follow traits through generations and try to figure out what's going on in genotypes and phenotypes. Also, a couple more things. The Punnett square can be very helpful for making predictions about, for example, children or for genetic diseases. So, for instance, there's a condition called albinism, an albino, where you see this. These are two sisters here, but you see this little girl has she lacks a pigment, melanin. So you see her hair lacks color, her eyes are blue, she has very pale skin, whereas her sister does not have that. So so we know that albinism, I guess I'm saying that right, is a recessive trait disorder, I guess you could call it. So you're going to use a lowercase a for the allele for it, and to be phenotypically an albino, you would have to be homozygous recessive for that disorder. So if you know that both parents are carriers, here are the two parents, and you set up a Punnett square, see the father's going to make half normal, half albino allele sperm, same with the eggs for the mother, fill in the Punnett square, you can answer the kinds of questions that people ask when they go to genetic counselors when they're having children. Well, if they know they're carriers or suspect they're carriers, they could say, well, what is my chance of having a baby that has albinism? And you would set up the Punnett square, fill it in, and notice that they have a 1 in 4 chance, or a 25% chance of having a baby that lacks the pigment or is albino. So there's many genetic disorders that are inherited in a recessive manner. Again, what does that mean? It means you have to have 2 recessive alleles, just like the white pea flowers could only be one, they had to be homozygous, and the recessive allele. The same is true for many genetic disorders. So, for example, I'm trying to think of a good example. Cystic fibrosis. Let's use that one as an example. Cystic fibrosis, you would be told in a genetics problem is that it's a recessive disease. So that's telling you that you have to have 2 defective copies for it. Okay, so I'm going to just, and you can pick your own letters just so you define them, but I'm going to use, let's see, cystic, I'll use an F. So lowercase f is for cystic fibrosis. Oh, I was talking about sickle cell. Sorry, let's go back to sickle cell. So sickle cell is also a recessive disease, so you have to have 2 defective alleles to have it. So this would be for sickle cell. The normal gene, we'll just say we'll use a capital S. Okay, so if you are, so to have cystic fibrosis, this would be your genotype, little s, little s. If you don't carry the allele at all, you'd have capital S, capital S. 2 normal genes. You're not expected to know that this disease involves the gene for hemoglobin, although later in Unit 4 I'm going to use that as an example when we talk about mutations, but for now we're just looking at the genetics of it. So if you're told something's a recessive disease, you got to have 2 defective copies of it, 1 from each parent. Now what if you have 1 good copy and 1 bad copy? Then, and it's a recessive disease, then you're called a carrier for that. So you yourself will be fine. You probably won't even know you're a carrier, but you do carry the defective allele. And the bad thing about that is that if you have children, you could pass it on to them. So here we see a Punnett square that shows the different possible outcomes if you're talking about recessive disease and both parents are carriers. See, they both have a normal and a defective allele. Normal and defective. So you set up the Punnett square, the sperm, normal and defective, same with the egg. Fill in the Punnett squares and then analyze it. And you see that they have a 25% chance of having an affected child. What about their other kids? Their other kid, 3/4 of that, there's 75% chance that they will have a normal, healthy baby. But you could add to that they also have a 50% chance of having babies that are carriers. Now, this does not affect their phenotype. So, usually, so they're considered phenotypically normal, but they are carriers. So there's lots of recessive genetic diseases: cystic fibrosis, sickle cell. You certainly don't have to memorize this, but this shows in the different chromosomes some of the different diseases. There's cystic fibrosis, certain types of colon cancer. There's certain types of breast cancer, polycystic kidney disease. There's sickle cell, malignant melanoma rather. And then I wanted to mention this one here, Huntington's. Huntington's disease is an interesting one in that it's one of the rare dominant diseases. It's a dominant genetic disease. In other words, all you need is to get 1 bad copy and you have the disease. So if you were doing a genetics problem with Huntington's, you would use a capital H for the Huntington's allele and lowercase h for the normal, and you would be told, this is an dominant disease. The others, like cystic fibrosis and sickle cell, were recessive. You had to have 2 bad recessive copies, but dominant disease, just 1 will give you the disease. So this person would have Huntington's, and so would this person if they had this genotype. They both have Huntington's disease. The healthy person would be little h, little h. So you do see some examples of that as well. Just so make sure you read your genetics problems carefully. To test for there's a test for many of these diseases, like sickle cell, like cystic fibrosis. They now have tests they can do, and they either do it by usually either by amniocentesis, or they'll take, we talked about this a little bit in an earlier lecture, they'll take a little bit of fluid. You see this needle here. They'll take a little bit of fluid from the amniotic sac around the baby that's going to have some of their cells in it. They'll culture those, and then we won't go through the process, but eventually they'll extract their chromosomes, and they'll do a karyotyping where they'll make sure the baby has all the proper chromosomes, and at this point they can also do genetic tests on them. There's also something called CVS sampling that they do now, chorionic villus sampling. It's the same idea. You want to get some of the baby's genes, chromosomes, their DNA, their genetic material, so you can test for some of these genes. And in CVS sampling, you can do it earlier than amniocentesis. It's a newer procedure, although they've both been around a while. But it's, you take a little snip of this structure that forms the placenta. It's called the chorionic villus. So you just go in intravaginally, so you don't have to stick a needle in. Intravaginally, take a snip of this tissue, and you can analyze the DNA. It works even though it's not actually from the baby's cells, but the placenta has the chorionic villi that are part of the placenta comes from the same cell that form the embryo and the fetus. So it's going to have the same genetic material in it. So you can use it to analyze the baby's genes. So they would do a karyotype like this that we've looked at before. Okay, one more thing. This is getting to be a long lecture here. Sometimes, most of the time, the alleles that we're talking about are not on the X or the Y chromosome. And in that case, we call those, I don't see it here, so I'll write it, autosomal traits. Or like if it's a disease, we'd say it's an autosomal disease. That means not on the X or Y chromosome. So autosomal means on any of the chromosomes that are not the X or Y. If the gene that we're talking about is on the X or the Y chromosome, we say that it's sex-linked gene. And most of them are going to be on the X chromosome, because the X chromosome is much bigger than the Y chromosome. It's got lots of genes on it. So sometimes they'll talk about X-linked traits and Y-linked traits. And there are some disorders that are sex-linked. They are on the X chromosome, so they're X-linked. Color blindness, Duchenne's muscular dystrophy, hemophilia, which is a blood clotting disease. These are all pretty, well-known X-linked alleles. Now, I'm bringing this up because you're going to do some genetics problems that are sex-linked, and you have to set those up slightly differently when you do your Punnett square. And by that I mean that not only are you going to use the letters for the alleles for whatever trait you're following, like big A or little a for whatever trait, some generic trait. So these are going to be your alleles, big A and little a. But, so if this was an autosomal situation or pair of alleles, you would just use those letters. You put like big A down the side, little a across the top. But because these are sex-linked, you also include the X and Y chromosome. And you put the alleles as a superscript. So you would say like the A allele is on the X chromosome. Okay, so the X chromosome can have the capital A allele or the lowercase a allele. And then the Y chromosome, notice it has nothing written on it, or no A or B, because if it's X-linked, it is not on the Y chromosome. You got to remember that. So don't put an A, big A or little a, on the Y chromosome, because if it's X-linked, that's exactly what they mean. It's linked to the X chromosome, not the Y. So notice in all these examples, the Y is there. You need it there to keep track of the allele, the chromosome, the alleles, but there's no, the Y chromosome doesn't have the A or allele written on it. So let's say you cross a female that's homozygous for the A allele, capital A allele, and then the male has the lowercase allele, and then the Y chromosome. So you'd put that across the top. If the gametes that this male would make, half would contain an X chromosome with a little a allele, and the other half of the sperm would contain the Y allele. And then for the female, well, there's only one kind of gamete she can make, the X chromosome with the big A, because she's homozygous. So you put that down the side, and then you just fill in your Punnett square like you always do. So that part's not different, it's just you have to include the X and the Y. And then when you go to analyze it, notice that you can say more here, in that you can not only can you give the traits of these offspring, but you can say whether they're male or female. So for example, let's say big A is normal, and let's use albino, and little a is the albino gene. Oh no, that's not X-linked. That's not a good example. All right, let's use hemophilia. Hemophilia is an X-linked disease. So we'll use little a for the hemophilia gene. So this female is normal. Okay, so her half of the offspring are going to be normal females, and then the males are going to be normal males. But look over here in this middle example here, if half of the X chromosomes carry a defective allele and the other are normal, and then that's crossed with a normal male, normal and the Y chromosome, then when you fill in the Punnett square, you see that if these are the parents, they do have a 1/4 or 25% chance of having a male baby with that defect. Say hemophilia, or this could be muscular dystrophy or something. And then notice in this third one over here, if the male carries the defect, the defective allele, and the female is the carrier, then it's going to be even worse. They're going to have a 50% chance of having a baby with that disease, and half of those will, it's a 50-50 whether it's male or female. And then they have a 1 in 4 chance of having a girl who's a carrier, and then they'll have a 1 in 4 chance of having a normal male. So those are sex-linked diseases, and you'll be practicing with those. So that completes our lecture on genetics.